Bismillahirrahmanirrahim. This is the June 2024 question paper 2-1. Second video, question 4, 5, and 6 are going to be solved in this. Question number 4, figure 4.1 is a scanning electron micrograph showing the tissues that line the bronchi and the gas exchange system. Figure 4.2 is a transmission electron micrograph of a horizontal section made at the position indicated by the two arrows in figure 4.1. So figure 4.1 have the transmission electron micrograph. 4.2 is a transmission, 4.1 is a scanning electron. So this one is the scanning electron and that one is the trans at the position indicated by the two arrows in figure 4.1. You can see the arrows here. Now the question here is here, and figure 4.2 magnification is 80,000. The magnification of 4.1 was 3,000. Name the cells labeled A and B. A was what? Ciliated epithelial. Ciliated epithelium. Epithelial cell. And B is the goblet cell. Right? So that's get you 2 marks. Then part 2 of the question says describe how the tissue shown in figure 4.1 is adapted to its function in the gas exchange system. Now 4.1 was this. Please look at 4.1 and then tell me how is it adapted for gas exchange, and that's 3 marks. Now what are you going to say for 3 marks? Goblet cells produce mucus, 1 mark. Mucus covers the epithelium to trap particles. Ciliated epithelium move the mucus upwards towards the throat. Very simple, very basic, 3 points, 3 marks. So the mark scheme is very clear. Goblet cells produce mucus. You could have said B cell secretes or produces or releases mucus. Mucus covers the epithelium to trap particles. You could also say to prevent pathogens from reaching the gas exchange surface. You can also say acts as a barrier to bacteria, or you could even say a named pathogen. And number 3, cilia move the mucus upwards towards the throat or towards the mouth, or you can say pharynx away from the gas exchange surface out of the airways. So 3 marks, 3 points. Then coming to the B1, the structures labeled X in figure 4.1 have a characteristic internal appearance as seen in figure 4.2. Describe the internal appearance of the structure labeled X. The structure labeled X is the cilia, and then they refer to 4.2, and 4.2 is this structure. This is the 9+2 structure, which is made up of the microtubules, and this magnification is 80,000 times. So then what would you write here? You'd write the 9+2 pattern of the arrangement, and this is composed of microtubules. So 9+2 arrangement composed of microtubules got you the 2 marks. Then part 2, explain how figure 4.2 shows that each of the structures labeled X are intracellular. Now let's have a look at a diagram which I want you to see, and then we can discuss this question. Now as you can see in this diagram, the label here is plasma membrane, and this is the plasma membrane. So this is the cell, and the cilia is outside. This is the cilia which is outside, and the inside part is called which is inside the cell, which is embedded inside the cell, is called the basal body. That has a 9-3 structure, and this one has a 9+2 structure. So 9 doublets and 2 in the center. So the cell membrane is enclosing it. Let me give it another color so that I can show you. This is the one, and this is the cell membrane. And then this is the cell membrane, which is continuous. So that's why it's mentioned here, plasma membrane. Now another diagram. This is the same sort of a diagram, but a little different. And you can see the electron micrographs of it. So this is the flagellum, which is slightly different, but it's longer. It's the same structure, but cilia are shorter and flagellum are longer, lengthwise they're longer. So you can see here again, we see the plasma membrane, and here again we see the plasma membrane, which is the cell membrane covering it. So each cilium is surrounded by a cell surface membrane, and that would get you your 1 mark. So contents are inside the cell. The basal body is inside the cytoplasm. Part C says stem cells are found in the lining of the bronchi. Describe the functions of centrioles and explain how they are involved in the cell cycle of a stem cell. So they've given you the question. They said stem cells are found in the lining of the bronchi, and describe the function of the centrioles and explain how they are involved in the cell cycle of a stem cell. So centrioles make or organize the microtubules so to form the spindle fibers, and during each cell cycle each centriole replicates, forms two centrioles. During S phase, G2 phase, the centrioles replicate, then centrioles move to the opposite poles, and centrioles then lengthen, or you can say shorten, the spindle fibers. You can't use the word contract, that's the wrong terminology, it's wrong biological English. So the question they had to talk, they were asking you about the centrioles. There's 6 mark scheme points, any four of those could have got you your 4 marks. So centrioles make microtubules to form spindle fibers. Each centriole replicates during, I usually teach it in the G2 phase, but S phase is also allowed. Centriole pairs move to the opposite poles. Centrioles lengthen or shorten the spindle fibers depending upon which stage of mitosis are we talking about. Question number 5. The pressure of water vapor inside and outside leaves can be measured. So this is a leaf, and inside it we can measure how much is the pressure inside and how much is the pressure outside. Say the pressure out inside was 50 and the pressure outside was 40. So the difference between these pressures is known as the leaf vapor pressure deficit, LVPD. So the difference here would be 10. LVPD is one of the factors that influences the rate of transpiration. Scientists measured the effect of changing the LVPD on the rate of transpiration in several species of flowering plants that live in a variety of different habitats. Two of these species were Nerium oleander, a species that is adapted to grow in hot, dry conditions. So this is the one which is adapted to grow in hot, dry conditions. And then Helianthus anus, a species that is not adapted for survival in hot, dry conditions. So Nerium oleander and Helianthus anus. Figure 5.1 shows the effect of increasing the LVPD on the transpiration rate of the two species. All other factors were kept constant. Now we have to look at a graph. Now graphs are always very, very technically difficult, and some of you are very good at it, some of you are pathetic, but then that's what all. We have a mixed bag of students. So you can see on the x-axis we have leaf vapor pressure deficit in kilopascals. So you have 0, 0.5, 1, 1.5, 2, 2.5, and 3. And then you have the two ones. One is, this is the Hale H, let's call it HA, and let's call this NO, Nerium oleander. Now you look at it, let's look at the next question. It says compare the results of the two species. Now please remember, if it's a describe or a compare, well then there's no biology. You're not going to give me any explanations. And these are the 3 marks which every student must get. But surprisingly, many of you don't get this because you write explanations and I don't want any bio. So this is no bio. Please remember, it's a compare of the graph. You're doing nothing but just comparing the graph. And we do not require any bio. You might not have even read this chapter and you don't know this chapter at all, but you can still do this question. So now, what are the points that you needed to give me? The first important point is you start, peak, end. Is a rule that we follow. start, peak, end. If the graphs, then you compare the start and the peak and the end. So no transpiration when LVPD is 0 kilopascals. You have to say the unit, 0 kilopascals. So no transpiration when LVPT is 0 kPa. Both increase in transmission until 2.5. You can see here both of them are increasing and then becomes constant. So both increase in transpiration until 2.5 kPa. That got you another mark. Then HA rate remains constant between 2.5 and 3. You can see here it remains constant. So HNUS remains constant between 2.5 and 3 kPa. So HA remains constant between 2.5 and 3 kPa. And the other one, Oleander rate decreases slightly. You can see there's a slight decrease here. If you look at it very carefully, there's a slight decrease here. So HA remains constant, NO rate decreases slightly. You must write the whole name. I'm not saying just write these abbreviations. Then HA has a higher rate of transpiration at all LVPDs. Then HA has a higher rate of increase between 0 and 2.5. So HA has a higher rate of increase, between 0 and 2.5. Between 0 and 2.5. And then you give me a comparative data quote. And what does that mean? I've talked to you in many, many papers of that. Comparative data quote means that I always say take one of the beginning. So at 1 kilopascals, give me the values of both of these, this here and this here, and you give me this value and you give me this units, and then you give them to me at the end. You could have done any two figures. I'm not saying you need to do only 1 and 3. You could have done 1.5 and 2.5 or something like that. And then you give me what were the figures at the end here, and you read off this on the y-axis. So data code means you give me units from both axes, and data must come from both the species, right? So 1 kilopascal, what was Nerium oleander, what was Helianthus, what was the rate for it? And then at 3, what was the rate? So this and this. And this gets you 1 mark. This is called a data quote. Data quote. Quote means quotation. So a comparative data quote from figure 5.1. Now I've given you all the points. I've given you, this is the entire data quote, but you have to write the units. The units are the most important thing. So you've got to give me the units on the x-axis and the units on the y-axis. Now no transpiration where LVTB is 0. Both increase in transpiration rate until 2.5 kPa, H annulus constant 2.5 and 3, H annulus higher rate at all LVPTs, and H annulus higher rate of increase between 0 and 2.5. So please try to look at the graph and then look at these points, and I am sure you all can develop the skill of writing this compare and describe and all these sort of no bio, which doesn't require any biology material that you should know. Then coming to the B part of the question, figure 5.2 shows part of a plant and oleander, and figure 5.3 shows a cross section of an oleander leaf. Now you can see here there are the 3 layers of the upper epidermis, similarly in the lower epidermis. Then you can see the trichomes, then you can see these grooves, and in which we have the trichomes, and these are the vascular bundles, and then you have many layered palisade mesophyll cells. Figure 5.3 shows a cross-section part of an oleander leaf. So 5.2 is the picture of the plant, and then it's the histology slide of the leaf. Now it says Figure 5.4 is a drawing of a high power of region N on figure 5.3. N on figure 5.3, this area. And it says state and explain two adaptations. State and explain two adaptations shown by the leaves of N. oleander that are visible in 5.3 and 5.4. So one adaptation visible in 5.3, explanation, one adaptation visible in 5.4, and explanation. So 5.3 is this one, and 5.4 is this one. Be very careful when you're reading the question. And this is going to get you 4 marks, and it's a total of 7 marks. Now, one adaptation in 5.3. Let's go back to 5.3. 3 layers of thick walled cells at the upper surface. What will that do? Reduces diffusion of water vapor to the atmosphere, which reduces cuticular transpiration. Then no stomata on the upper surface, stomata only on the lower surface, stomata not exposed to direct sunlight. Then this will be 2 points. Then 5 stomata in pits, or you can say in cavities or depression or infoldings or grooves. Many words you can use. It depends on how good your English vocabulary is. And this creates a humid atmosphere in the pit, so it reduces the diffusion of water vapors. Then figure 5.3 or figure 5.4, you could have said a thick-walled epidermis reduces diffusion of water vapor through the lower epidermis. Then you could have said thick waxy cuticle on the epidermis, so it reduces the diffusion of water vapor through the cuticle. Then you could have said the trichomes or the hairs around the stomata, and this reduces air movements. Or you can say it traps still air, which will create a humid atmosphere. So will reduce the rate, the diffusion, or will reduce the diffusion or the potential water potential gradient for water vapor. So lots of points you could have given me, but it depends how sensibly you answer this question. So 3 layers of thick walled cells at upper epidermis reduces cuticular transpiration, hairs, trichomes around the stomata, reduces air movement, traps still air. Please go through the mark scheme, and then you can see the other points which were also available to you, which I have of course discussed verbally with you, but I have not written them down. Question number 6, this is the last question. Antibodies are produced by plasma cells. Figure 6.1 shows antigens bound to antigen binding sites of an antibody molecule, and you can see it's the label antigen and then hinge region, light chain, heavy chain. Explain how the structure of an antigen binding site makes it specific to a particular antigen. Figure 6.1. Antigen binding site has a variable region and has a shape that is complementary to the antigen. The variable region has specific sequences of amino acids, which is the primary structure, and different amino acids have different R groups, so give different tertiary structure and shapes for binding to antigens. So mainly it's the primary structure or the amino acid sequence which will dictate the shape of the antigen binding site or the variable region. So complementary to antigen specific primary structure, different amino acids have different R groups, so will give a different tertiary structure. Then it says part 2, state the function of the hinge region of the antibody shown in figure 6.1. Now basically what is the hinge for? On a door, what is the hinge for? So allows flexibility for binding to antigens, or you can say it allows the variable region to move. So that is the 1 mark. Then it says antibodies can bind to membrane receptors on cells of the immune system such as macrophages. Suggest an advantage of antibodies binding to receptors on macrophages. Like for instance, this is a macrophage, and these antibodies can bind here, there are receptors here. Now, it says suggest an advantage of antibodies binding to receptors on the macrophages. Macrophages are the phagocytes. So, easier for macrophage to engulf because the antibodies that have found the antigens or engulf the antibody antigen complex. And this will facilitate the destruction of the pathogen which is marked by the antibodies. Because these antibodies, you see, this is the virus, and it's got these antigens on its surface. Now I'm going to give it another color. So these antibodies bind to it, right? And then here comes our hero, the Superman. And this is the phagocyte. And then it has receptors to which these antibodies bind, and then it's going to engulf, and that's going to be a phagolysosome, and that's going to digest this pathogen. So what you've got to understand is why is it important to suggest an advantage of antibodies binding to receptors on macrophages. So easier for the macrophage to engulf the antibody antigen complex and facilitates the destruction of the pathogen, so the person is not going to suffer from the disease. So I've given you the points for the part 2 allows flexibility for binding to the antigens, and then in part 3 the one mark was facilitates destruction of the pathogen marked by the antibodies, and the other was easier for the macrophage to engulf antibodies that have bound to the antigens. Part B says it is estimated that the immune system of each person can make enough antibodies to bind to over 10 raised to power 12 different antigens. When plasma cells make antibody molecules, they combine the polypeptides produced by the expression of genes for heavy chains and the genes for light chains. Research has shown that producing this very large number of antibodies is only possible by modifying the primary transcript of the genes that code for heavy chains and the genes that code for light chains. Suggest how this modification of the primary transcript occurs in plasma cells. So removal of introns from the primary transcript, then when you've removed the introns and the exons are joined together in different sequences, in different combinations, which is called alternative splicing. Not all the exons are used in making the polypeptides. So removal of introns, 1 mark, exons are joined together in different combinations. Not all the exons are used in making the polypeptide. So you were only supposed to give me 2 points how this modification of the primary transcript occurs in the plasma cells. So this is of course new in the syllabus and was put in it 2022, so we don't have many questions on this, but everybody must know this, and it has come in quite a few of the papers which have come in the recent 2 years. So 22, 23, 24, we have 3 years of papers which we can go through. So that finishes this paper. Thank you very much.